Hi,
Hi.
uh, welcome to Science Hangout from Science Mission. We have today Dr. Ted Price from the University of Dallas, where he is the associate professor at the School of Behavioral and Brain Sciences. Dr. Price is a recipient of Young Investigator Awards from the American Pain Society and International Association for the Study of the Pain. Dr. Price has published more than 50 peer-reviewed articles in international journals. He serves as the pharmacology section editor for European Journal of Pain and is on the editorial board for Pain, Molecular Pain, and the European Journal of Neuroscience. He is also a regular reviewer for National Institute of Health and American Pain Society study sections. Welcome, Dr. Price, to Science Hangout.
Hi. Thanks for having me.
So, can you please tell about yourself and, excuse me, your background, research interest, education, etc.?
Yeah, sure. So I did my undergraduate work here at the University of Texas at Dallas
Mm-hmm.
in the 90s in neuroscience. I then went to
Okay.
the University of Texas Health Science Center in San Antonio to do a PhD in pharmacology and neuroscience.
Okay.
When I was there, I started working on pain with Chris Flores and Ken Hargreaves.
Okay.
And after I completed my PhD, I went to McGill University to work with Fernando Cervero,
Uh-huh.
staying in the pain area, and had a really wonderful experience at McGill University in the Center for Pain Research that they have there, which was at the time one of the biggest pain research centers in the world, and still is. And then went to University of Arizona in 2007, where I
Mm-hmm.
started my independent lab. I was in the Department of Pharmacology in the medical school.
Uh-huh.
Started my lab and worked there for 7 years, and then 2 years ago came full circle and came back to the University of Texas at Dallas.
Mm-hmm.
So I'm now an associate professor here and the director of the undergraduate neuroscience program, which is interesting because I was in the first graduating class for this program back in 1997. So it's a lot of fun to be back here. The university's changed in tremendous ways, and I think that we're doing some pretty exciting stuff here.
Okay. And what made you just think about just only academics, not industry? Because there is a lot of work going on in industry too on pain research.
Yeah, that's true. So when I first started, when I was doing my PhD, I was considering both an academic career and an industry career. And
Uh-huh.
I think that really what happened is that about the time I was finishing my postdoc,
Mm-hmm.
a lot of the industry jobs started drying up. So I
Okay.
finished my postdoc at about the same time of the financial collapse of
Okay.
2007 and 2008.
Uh-huh.
Got to witness firsthand almost all of the people that I knew that were working in basic science in pharma
Uh-huh.
get laid off, and
Okay.
that was frightening. So
Mm-hmm.
up until about 2007, 2008, I think most people thought that industry positions in basic science in pharma were actually more stable
Yeah.
than academic positions, and I don't know anybody that thinks that anymore
Yeah.
for good reason. I mean,
Yeah.
it's really hard. It's really difficult to maintain a program in basic neuroscience, especially in pharma nowadays. And I actually think that both careers are extremely interesting. So I'm interested in drug discovery. My lab is
Mm-hmm.
interested in drug discovery.
Mm-hmm.
That's what we're doing, and I think it's more and more people now are doing that in academia.
Yep.
But doing it in industry is also very attractive. There are a lot of resources. It's just, I don't think, a very stable position anymore unless you're at the very top. And coming out of a postdoc, you're not really looking at going into at the very top.
Yep.
So I think being an academic scientist is a wonderful career.
Uh-huh.
I love teaching. I love interacting with students.
Mm-hmm.
But I think at the end of the day, the reason that most of us go into this is because of the research.
Yep.
And the research opportunities in academia and industry are both quite exciting. So it was a tough decision that was mostly made for me by circumstances that were completely out of my control and maybe out of anybody's control.
Yeah, that is good. I mean, like, at least you had it at the right time to make that decision, and it happened at the. I mean, like, sometimes you feel it difficult to make that decision.
Yeah. So it was a difficult decision to make, and I was lucky that I had an opportunity to potentially do both, and I'm very happy that I'm in academia. I'm also interested in commercializing what we do in the lab. I'm not going to talk about that today specifically, but we have started a company out of work that we do in the lab. It's based here in Dallas, and I think it's really exciting.
Yeah. That is good. I think a lot of work goes out. I think most of the patents get commercialized nowadays from the universities, which is a good opportunity for the faculty to look into it and get something out of it, like as you said, like by doing your own, starting your own startups and all those things.
Yeah, absolutely true.
Can you please talk about your mentors or supporters who helped you along the way?
Yeah, sure. So my first mentor was here at University of Texas at Dallas, Allison, when I was a junior. Had just switched from a physics major to a neuroscience major. And, started working in her lab doing face perception studies, which is something that I haven't done since I worked in her lab. But she was just a fantastic mentor in terms of thinking about how to do science and how to design meaningful experiments, how to analyze data. And, she taught me a lot about what to look for in mentors. And I was really lucky to continue to have great mentors down the line. So when I was doing my PhD, I worked, like I mentioned, with Chris Flores and Ken Hargreaves. Chris Flores is now at Johnson & Johnson. Ken Hargreaves is still at the University of Texas Health Science Center in San Antonio. And they were just tremendous mentors, teaching me how to write, how to think about putting together projects. The two of them ran a grant writing class, which I don't think I appreciated as much as I should have when I was a graduate student, but I certainly look back on it a lot now. I think, those lessons that they taught me were really very important in helping me develop my independent career. And then when I was in Fernando Severo's lab at McGill, Fernando was just an exceptional mentor. And he was an exceptional mentor, especially in helping me think about how to position the science I was doing within the field, how to think about building projects in relation to what other people are doing, which was something I hadn't really considered before. But I think has also played a really big role in helping me to be successful. He also broke me of the bad habit of writing very long sentences, which was important. And then, I got to University of Arizona, and again was extremely lucky to have a faculty mentor, Frank Parecka, who took me under his wing, helped me to really refine the things that I still needed to work on, which I think was developing focus and understanding how to look at projects rather than on a 1 or 2 year window and a 5 year window. And, he really helped me to get the funding that I needed to get the lab started. He was great in helping me to recruit people to my lab, how to think about recruiting the right people to the lab. And we started to build a really great group of people, which we've continued here. So, mentors play a really, really important role in developing your career, and finding the right mentors is really important. And I've been really exceptionally lucky, starting from the time I was about 20 years old to work with just wonderful people who have put my career interests at the forefront.
That is nice to know. I know you are busy with a lot of work and lab and other things. So, if at all you get some time, what do you do? What are your hobbies?
Well, I have a 1-year-old baby girl,
Okay.
Penelope, and
Okay.
I try to spend as much time with my wife and her as I can.
Uh-huh.
So that has become my primary hobby, is being with
Okay.
my very young daughter. But when I'm not doing that, I try to play basketball as often as I can. So
Okay.
that's my favorite thing to do when I'm not working,
Okay.
or when I'm not with my daughter. So every day at lunch, I try to get over to the gym here at UTD and play basketball with a bunch of people half my age that run circles around me.
Uh-huh.
Yeah, so that's how I try to act like I'm still young.
Yeah, I mean, like this is a good place for basketball, I guess, with the
It is.
Dallas Mavericks and all.
Yes, that's true. Yeah.
Yeah. And let's talk about your lab. So, can you please give an overview of lab, how it works in your lab, grad student, postdocs, undergrads, and all?
Okay. So the lab is run by 2 PIs. So there's me and Greg Dusor. Greg and I did our PhDs together in San Antonio in Ken and Chris's lab. We went and did our postdocs at different institutions, and then both got hired at University of Arizona at almost exactly the same time. And for the last 9 years, we've run our lab together. So that's the kind of over structure. And then right now we have 7 postdocs and 6 graduate students, and a lot of really talented undergraduates in the lab. And basically the way we run the lab is that we have postdocs and graduate students that have their own projects that are usually split off of the grants that we have. And then they assemble teams of undergrads that they recruit and that work with them. And so they act as kind of the primary daily mentor to the undergrads that work on their team.
Okay.
And we obviously are as involved as much as we can be, but we're primarily interacting with the postdocs and graduate students. So we have lab meetings once a week where we hear about what everybody's doing or what they've been doing over the last week or two, and try to interact with people down in the lab as much as we can. We just moved into this beautiful new building here, which we came into in February of this year. So we're in this just absolutely amazing research space here, and all of the biology and neuroscience labs on campus that used to be spread all over the place are now in one building, or 2 buildings actually, but they're connected by bridges and tunnels, and they're right next to each other. And it's really a fantastic intellectual atmosphere, and I think it's really enhancing our ability to build new collaborations, and creates a fantastic atmosphere for our graduate students and postdocs.
Okay. Now that you mentioned your lab, let's look at some of your work, if you don't mind. Can you present your work?
Yeah, sure. So let's go to this. Did that work?
Yep.
Okay, great. So what I'm gonna talk about today is a couple of published papers and some unpublished work that we have been doing. And this is something I presented at the American Pain Society meeting down in Austin, Texas, with some changes about a month ago. So what I'm going to talk about is switching circuits, how differences in coding of acute and chronic pain may lead to better pain therapeutics. So the first thing I want to do is just show my research group here. So does the cursor work? Yep. Okay, great. So I mentioned that this is a collaboration with Greg Dusor. This is Greg right here. And then the people that worked on the project I'm going to tell you about today are Ji-Young Kim, who was a PhD student who was in our labs when we were at University of Arizona, who finished and is not in this picture. Jamie Moy, who's a senior PhD student in the lab, and Salim Megat, who's a postdoctoral fellow who's been in the lab for about a year and a half now. But this is the whole group as of I think this picture is about a year old. So we have some new people that have joined the lab that aren't shown here, but this is the group overall. Okay. So we are interested broadly in pain, but in particular, we're interested in chronic pain. So this is a painting that I like to show to demonstrate the differences between acute and chronic pain. So the name of the painting is A Stab in the Arm. It's by a gentleman named Stan Cohen who lives in Oakland, California. It's part of an online art exhibit called Pain Exhibit, where you can see art produced by a large number of artists that have suffer from chronic pain. So if you imagine that you were just stabbed in the arm, so what do you do when that happens? You reach for the injury, you protect the injury. The nociceptors, the sensory neurons that innervate the area, become active. They're shown in red here, so that demonstrates that they're firing action potentials. They're red and angry, and they're sending a signal into the central nervous system that enters in the spinal cord, shown here, and then moves on up to the brain to tell you, something terrible just happened to you and that you need to protect yourself from it. So you need to get away, first of all, from whatever it was that hurt you. And number two, if you have some lingering injury, which if you just got stabbed, you obviously would, you need to protect it during the healing process. So acute pain serves an important function. It protects you from things that can hurt you. We know of genetic diseases in human beings where people fail to develop nociceptors. They can't feel pain. In people that have those diseases, they usually have lost fingers and toes, sometimes entire limbs by the time they're a couple of years old. They're not able to protect themselves, and they don't live very long. So it's really important to be able to feel pain. It's a vital protective function.
Okay.
It is a nuisance when you're healing from something and the sprained ankle or whatever is getting close to healing. You want to get back out and do what you want to do, but it still hurts. That's happening because it's protecting you from re-injury, letting the healing process run its entire course. And in most cases, that goes back to normal, and you don't realize that months after, you don't realize that anything ever happened. Well, that's not always the case, though. So in this particular case, Mr. Cohen was not actually just stabbed in the arm. What actually happened was that about 10 years prior, he had an injury to his cervical spinal cord and had a surgery to repair it. So even though he had that repair to the spinal cord, and the spinal cord seems to be perfectly normal, he still has pain, like he's being stabbed in the arm all the time. Okay. So what happens in that case? Well, I think that we now have pretty good evidence from preclinical animal studies and clinical studies that the nociceptors that innervate the area are sensitized and probably still firing action potentials, even though there's no injury evident anymore. Okay, so that plasticity that caused those nociceptors to become hyperexcitable never went back to normal. Okay, so there's some persistent form of plasticity that did not reverse. And in the spinal cord, there's amplification of signals as they come in and get sent onto the brain. We sometimes call that central sensitization. And that also has persisted past the time of the injury, in this case, almost a decade, right? So this is clearly maladaptive now. It's not telling your body that you need to protect yourself from an injury. The injury's gone. It's not telling you that you need to protect yourself during a healing process. The healing process has long since finished. So there's some form of aberrant plasticity now that is driving this chronic pain state. And ultimately, what we'd like to be able to do is to reverse that. So this next slide shows that. So we have our normal protective pain, as I was mentioning. Acutely, it leads to reflexes. So like if you touch a hot stove, you will withdraw your hand from the stove. If you leave it on there a little bit too long, you get this prolonged pain from the inflammation that you get from the mild burn injury, and you may hurt for a couple days, but with any luck, it goes back to normal. However, in some number of cases, you have this abnormal, non-protective pain that persists after the injury resolves, or can even come on after there was no apparent injury whatsoever. So migraine headache, which Greg Dussor spends most of his time studying, is a good example of this. And migraine headache, we really don't know what precipitates it in the first place, what causes it to happen. But people have these pain episodes. They can have pain episodes for decades of their life. Mm-hmm. And we'd like to be able to understand where this plasticity is happening in the nervous system. So that has led to this idea of chronic pain as a disease. So we look at chronic pain as a disease of plasticity. It's a disease of the either peripheral or central nervous system. That's something that I'll try to give you some evidence that it's a disease of a particular part of the central nervous system today. And we'd like to therapeutically come up with molecules as targets that would allow us to eliminate this abnormal pain without interfering with this normal protective pain that we all rely on to navigate our environments. So how do we do this? So we use a number of different models in rodents. This is the one that my lab uses most frequently. This particular model is called a model of hyperalgesic priming. So I'll explain briefly how this works. So this was developed by a guy named John Levine, who's at the University of California at San Francisco in the late 90s and early 2000s. And he's done a ton of work on peripheral mechanisms of hyperalgesic priming. So the model basically works like this. So if you take an animal and expose them to an inflammatory mediator like prostaglandin E2 at a relatively low dose, that causes a very short-lived pain response or no pain response at all. However, if you have previously submitted that or exposed that animal to an inflammatory stimulus like carrageenan or interleukin-6 or nerve growth factor, which are things I'll talk about in the talk today, and then come back after these animals no longer show any pain response, and expose them to PGE2, you now get a very long-lasting pain state. So the idea here is that the animals have become primed by this initial stimulus, and then when you give them PGE2, that PGE2 causes a very long-lasting pain state. So during this maintenance phase, we have an opportunity to understand what the molecules that are involved in causing this acute pain to transition to a chronic pain state. And we can intervene with treatments to potentially reverse this. So we can make the animals unprimed now. So we can go from this chronic pain state back to this simply acute protective pain state. So John Levine developed this model, which more and more people in the field are using now. He focused mainly on the peripheral nervous system using treatments that were targeting dorsal root ganglion neurons. This shows a little bit about the molecules that they've discovered. But as we were doing work on this, we became interested in plasticity that happens in the central nervous system, and really have spent most of our time focusing on how plasticity in the central nervous system can maintain these chronic pain states in hyperalgesic priming models. So if you're interested in the peripheral part, I would encourage you to go look at John Levine's work. This review published in 2009 is a really fantastic place to start. But what I'm going to do today is to focus on the central nervous system component that our lab has been working on. So our question then really is, what is the circuit that maintains chronic pain state in hyperalgesic priming models? So we had a couple of different ideas about where that circuit may be, and we used mostly toxin approaches to manipulate these circuits to discover if there are differences in circuits that regulate pain plasticity in acute pain versus once pain is transitioned into a chronic state. So one group of neurons that we focused on were NK1 positive dorsal horn neurons. These are neurons that express a receptor for substance P, which is a neuropeptide that's released by dorsal root ganglion neurons. These guys are mostly projection neurons that are found in lamina 1 and 2 of the spinal cord. So these are like the neurons that are at the interface between the peripheral nervous system and the central nervous system. They send a signal on up to the central nervous system for the spinal cord. And they're also modulated by local circuits that are found within the spinal cord and descending circuits that are coming back down into the spinal cord from the brain. Another area where we looked was at descending modulatory circuits coming from a place in the brain called the rostroventromedial medulla. The reason we focused on that is that this is an output center for the endogenous opioid centers and endogenous cannabinoid centers that are found in the periaqueductal gray. So there's been a tremendous amount of work done on these circuits and their potential role in pain chronification. And then we wanted to look at whether maybe something else altogether was involved. And I'll get in to the end, at the end, about a dopaminergic circuit coming from the hypothalamus that we think is actually the key to this. So as I mentioned, our first target was these NK1 expressing neurons in lamina 1 of the spinal cord. So this is a paper, these are some images taken from a paper that was published by Pat Manti and Don Simone back in the late 90s or early 2000s, where they used this approach that was has subsequently been termed molecular neurosurgery. So it's a really neat approach. What they do is they take a ligand for the receptor, in this case substance P, and they conjugate it to a toxin called saccharin. So saccharin is a ribosomal toxin. Under normal circumstances, it can't get into cells, but when it's conjugated to substance P, when substance P binds to the NK1 receptor, the NK1 receptor gets internalized. It acts then like a Trojan horse. So the substance P and saccharin comes into the cell. The saccharin can be released from the substance P. It binds to ribosomes and it kills the cell. So you can use this to eliminate the NK1 neurons that are found in the superficial part of the dorsal horn. So you can see that here with substance P saccharin treatment, and this is from Pat Manti's paper, many of these neurons are lost. You can see it in more detail in this blown up picture here. So what happens behaviorally when you do that? Well, the effect is really quite profound. So if you cause inflammatory pain of a variety of different types or neuropathic pain to animals where you've eliminated these neurons, you see a profound decrease in mechanical hypersensitivity, ongoing pain responses, or mechanical hypersensitivity after nerve injury, for instance, shown here. So these neurons, if you eliminate them before an injury, are required for the pain hypersensitivity that happens in response to the injury. So what we wanted to do was to ask a really simple question. Are these neurons, which we know are required for the pain plasticity to occur during the initiation phase, also required for pain amplification once pain becomes chronic? So we designed experiments like I'm showing right here. So we wanted to look either at the initiation of the priming or at reversal of the priming during the maintenance phase. So we could give the toxins directly into the central nervous system via an intrathecal injection into the spinal cord before we gave the inflammatory stimulus. And most of the data I'll show in the rest of the talk will be using interleukin-6, although we've also done most of these experiments with carrageenan. And this work was published in 2015 in Journal of Neuroscience. Ji-Young Kim was the first author on the paper. And then we look at what happens with priming when we challenge the animals with PGE2. So what we saw when we eliminated the neurons, it doesn't do the animation. So how would I do that? There we go. Okay. So removing the animation on the fly. So what we do is to give the toxin before we give the IL-6, and then we measure the response to the IL-6, and then give PGE2. So remember, I showed in the data from Pat Manti and Don Simone that if you remove these neurons beforehand, it removes the pain amplification response. And that's exactly what we saw. So what we're showing here on the y-axis is the mechanical threshold. So these animals develop a mechanical hypersensitivity. It's the pain plasticity that we measure. With IL-6 and the ones that were treated with the control, they develop a robust mechanical hypersensitivity, returns to normal, and then when we challenge them with PGE2, they are primed. So they have this persistent form of pain plasticity. In the ones that were treated with substance P saccharin, they have only a transient decrease in their mechanical threshold, comes back to normal very rapidly, and there's no priming. So these neurons are required for the establishment of this form of pain plasticity. So the next question is, are they required for the maintenance? So here we do the experiment in a different way. Rather than giving the toxin before, we give it after we've established the priming. So now the animals are primed, we give the toxin, and then we measure whether they respond to PGE2. And what we saw here was completely the opposite of when we gave it before. So in the animals that were treated with the substance P saccharin, they have a very robust response to the PGE2, whether they were primed with IL-6 or carrageenan. So we think that there's a fundamental difference in the neural circuitry in the spinal cord that's involved in the acute pain response versus the chronic pain response, where you no longer need these superficial dorsal horn NK1 expressing neurons for the maintenance of a chronic pain state. So that got us interested then in looking in these descending modulatory centers from the rostroventral-medial medulla, or the RVM. So these guys are modulated by the periaqueductal gray, which is shown up here. The periaqueductal gray is the place in the brain where most of the, or not most, but a lot of the endogenous opioid peptides are found, as well as endogenous cannabinoids. And when you activate the endogenous opioid or endogenous cannabinoid system, it stimulates the RVM and causes these neurons to release neurotransmitters in the spinal cord that modulate incoming pain information. So one of the neurotransmitters that is released here is norepinephrine. Norepinephrine is thought to act mostly as an inhibitory neurotransmitter in the dorsal horn. Another one that can be released here is serotonin. Serotonin seems to play a role in pain facilitation. So because we're mostly looking at pain facilitation, we decided to focus on serotonin, although we also did work with norepinephrine, and that I'm not going to show that data, but it was all negative, number one, and number two, it's all published in this paper. So let's go through what happened with serotonin. So with the serotonin toxin given intrathecally, we see that we had an elimination of the serotonin projections that are coming from the brainstem and the spinal cord. Mm-hmm. And when we look behaviorally, we see that the initial response to interleukin-6 or carrageenan is decreased. And then when we look at the priming, there is a small but significant decrease in the duration of the pain plasticity. So this suggests, like what we saw with the NK1 neurons, that descending serotonergic modulation plays a role in the initiation of pain plasticity and hyperalgesic priming. Mm-hmm. So again, we wanted to look at this in the maintenance phase. So now, rather than doing the toxin injection prior to the priming, we do it after the animals have already been primed, and we see if it has any effect on the plasticity. So here we're giving the IT injection after the animals have returned back to normal from the IL-6 or carrageenan. And again, like what we saw with the substance P saccharin, we see that this has no effect on the priming. So again, there's a difference between the circuitry that's involved in the initiation of pain plasticity and the maintenance of pain plasticity. So we think that this is, again, good evidence that the circuits involved in maintaining a chronic pain state are different than those involved in initiating acute pain plasticity. So there are a limited number of projections that come from the brain down into the spinal cord. And we felt like we had looked at the two ones that people had investigated the most. And those are these descending noradrenergic or serotonergic inputs that are coming from the RBM. So we shifted our focus to this less studied but very interesting hypothalamic nuclei called the A11 nucleus. So there are quite a few nuclei in the brain that contain dopamine. The most famous one, of course, is the substantia nigra. Another very important one is in the ventral tegmental area that projects to the nucleus accumbens. So the substantia nigra dopaminergic neurons, of course, project to the striatum and are very well known for their involvement in Parkinson's disease. These neurons that are found in the VTA that project over to nucleus accumbens are very well known for their role in the reward pathway. So these neurons that are found in the A11 nucleus, rather than projecting up into areas of the brain, project caudally down into the spinal cord, where their function actually is not very well understood. So we had the idea that maybe we could lesion these neurons and we would see some effect on the maintenance of hyperalgesic priming. So the approach was to use this very well-known dopaminergic toxin called 6-hydroxydopamine. And rather than inject it into the brain like people do to induce Parkinson's disease in animal models, for instance, we injected it into the spinal cord so that it would only have an effect on these A11 neurons. And this is showing these A11 neurons look like with the sham toxin. And this is what they look like with the toxin. So clearly we eliminate most of these neurons when we inject the toxin into the spinal cord. And what we saw in other dopaminergic centers is that this appears to be very specific for the A11. We don't, for instance, lesion neurons in the substantia nigra or in the VTA. So what happens behaviorally? So here, if we give the toxin before we do the IL-6 or carrageenan injection, we don't see any effect on the acute response. But different than what we saw with all of the other models, the priming is not there, right? So the animals show their acute pain plasticity, but they don't seem to transition to a chronic pain state. So this priming now is absent. So we wanted to do this the same way that we did the others, where we would do the toxin and eliminate the neurons after we'd established the plasticity to find out if these neurons are also involved in the maintenance. And indeed, they are. So when we eliminate these neurons after we've induced the plasticity, we still lose the primed state. So these neurons seem to play a key role in both the initiation and the maintenance of this chronic pain plasticity. We then wanted to understand which dopamine receptors were involved in this. So there are 5 different types of dopamine receptors. There are the D1, D5 receptors. They're expressed throughout the brain, also in the spinal cord. And then there are D2, D3, and D4 receptors. These are difficult to distinguish pharmacologically, so they're usually grouped as D1, D5 because there are drugs that target both D1 and D5, but you can't distinguish them. And then D2 and D3 usually are grouped together as well. So we used a number of D2, D3 compounds, which are, again, published in this paper. They didn't have any effect. But D1, D5 agonists had a very interesting effect. So when we gave a D1, D5 antagonist at the same time as the IL-6 or carrageenan injection, they didn't do anything. But once the pain had transitioned to a chronic state, they now were active. So we had a drug that lacks activity for acute pain, but very efficaciously blocks chronic pain. So this is pretty nice, right? So it would be a target that potentially would have no involvement in the normal protective pain, but could eliminate this aberrant pain plasticity. So we also wanted to see if D1, D5 agonists would have a specific effect in animals that had transitioned to a chronic pain state. So if we inject a D1, D5 agonist in naive animals, it doesn't do anything. It also doesn't cause them to grimace. Believe it or not, when mice are in pain, they make faces like they're in pain. If you inject this compound intrathecally in animals that have been primed, it does cause a pain state, and it also causes them to grimace, right? So suggesting that not only does it involve a change in the reflexive behavior, but also causes an effective pain state. So we're now really trying to focus on which particular receptor is involved in this. So D1 receptors are found throughout the brain. They play a key role in motor coordination in the striatum. They also play an important role in reward pathways. But D5 receptors are not expressed in many areas of the brain, and their function is not very well known. But interestingly, while D1 expression is relatively low in the spinal cord, D5 receptor expression is very high. So I'd like for everybody to kind of note where you see this expression here. It's the dark brown is where these neurons are. And they're mostly found in this medial portion of the dorsal horn here. So what Salim Magat has been working on in the lab is trying to find which neurons are engaged by the dopaminergic system when pain becomes chronic. And lo and behold, what he finds is that those neurons seem to be mostly here in this medial portion of the spinal dorsal horn. So if we go back to that previous slide, you can see that these are where there's really a lot of D5 receptor expression. So we're very excited about this because it suggests that we could potentially make a D5 receptor specific antagonist that would not have effects on the motor system in the striatum, may not have effects on the reward pathways in the nucleus accumbens, but could have a profound effect on pain plasticity in the spinal cord. So I think that most people would not think of D1, D5 antagonists as good drug targets, but if we could make a compound that was D5 specific and target these neurons that seem to be engaged only once pain becomes chronic, we could have a really good therapeutic option. So now what I'm going to do is change direction just a little bit and try to explain maybe how these dopaminergic neurons in the spinal cord are integrating with the spinal circuitry. So this is a figure from a paper that Michael Gold, who's a very well-known researcher in the pain area, published back in 2008, showing that in chronic inflammation, there is a change in the valence of GABAergic circuitry or GABAergic signaling in the spinal cord. So I think most people, even passingly familiar with neuroscience, know that GABA is the major inhibitory neurotransmitter in the brain. Yeah. In fact, under most circumstances, GABA is inhibitory, but under certain circumstances, GABA can become excitatory through a process that is now usually called disinhibition. So there has been a lot of work in the pain field on this idea that once pain becomes chronic, GABA becomes disinhibitory. So it can have some excitatory role. And this paper shows some pharmacological evidence for this. So what Michael's lab used was this compound called muscimol, which is a GABA-A agonist. And they show that in animals that don't have inflammation, muscimol is a really good analgesic. So if you give it into the spinal cord, it produces analgesia in a dose-dependent fashion. However, if you take animals that have pre-existing inflammation and you inject muscimol, it causes an exacerbation of the inflammatory pain at many doses. If you take the dose up high enough, it becomes analgesic again. But at some doses, it causes a disinhibitory exacerbation of pain. So we wanted to look into whether this was happening once pain becomes chronic in our hyperalgesic priming models. So we did some experiments where we gave muscimol at the same time of IL-6 or PGE2. And what we saw was quite striking. Like Michael Gold showed, very acutely, muscimol is a good analgesic, so it reduces IL-6 induced mechanical hypersensitivity. But if we give it in primed animals, it doesn't do anything. It's now completely ineffective. If we use a GABA-A positive allosteric modulator, midazolam, this is an effective analgesic again for acute pain, but once pain transitions to a chronic phase, it's also ineffective. So what about GABA-A antagonists? So if you take a GABA-A antagonist and give it to animals that have been primed, it causes a decrease in sensitivity. So this is the opposite of what it does in naive animals. If you give it to a naive animal, it causes pain hypersensitivity. Not only that, it blocks the grimacing that I told you about before. So it blocks this effective pain response that mice show when we challenge them with PGE2 after they've been primed. So there's this inversion of the valence of GABAergic signaling after pain becomes chronic. So not only that, but if you take mice, so here again is showing what I mentioned before. If you have unprimed mice and you give them a GABA-A antagonist, it causes pain. If you take primed mice and you give them a D1, D5 agonist, it causes pain. If you take primed mice, give them the D1, D5 agonist, and give them the GABA-A antagonist, that blocks pain. Okay, so there appears to be some connection between dopamine and GABA, and we don't think that's a coincidence, because if we go back to this slide, these neurons here that are becoming active only once pain becomes chronic, that express the D5 receptor, so these guys shown here, these neurons down here are mostly GABAergic neurons. So we think that these are GABAergic neurons that are projecting up into either the lamina 1, lamina 2, or down on these more superficial or deeper neurons found in lamina 5 that are typically referred to as wide dynamic range neurons. So with this in mind, we wanted to look at changes in molecules that modulate GABAergic synapses. And we focused on two things. We focused, number one, on KCC2, which is a potassium 2-chloride transporter. So the potassium chloride transporter extrudes chloride. KCC2 extrudes chloride from neurons. It's involved in maintaining a low intracellular chloride concentration. And then we also looked at synaptic adhesion molecules called neuroligans. We focused on neuroligin 1 and neuroligin 2. The reason that we did that is neuroligin 1 is found at excitatory synapses, so glutamatergic synapses, whereas neuroligin 2 is thought to almost exclusively be at GABAergic inhibitory synapses. So we looked at whether the expression of these things was changing as pain became chronic. And what we saw is that when pain became chronic, there was no change in neuroligin 1 expression. There was no change in neurexin 1 expression. So this is the ligand for neuroligans. Surprising to us, we didn't see any change in KCC2 expression. This does very robustly decrease with neuropathic pain. So, there may be different mechanisms for different kinds of chronic pain, but neuroligin 2 expression was increased. So this suggests that there's some change in the architecture of GABAergic synapses in the spinal cord. So at about the same time that we were doing this work, a paper came out describing a peptide called neurolide 2 that interferes with neuroligin 2 function. So this was discovered by another group. It was published in Neuropsychopharmacology. But we thought, hey, this is a really great opportunity to try to disrupt neuroligin 2 function with this compound and see if that changes chronic pain states. So what we did was first to show that neurolide 2 did what it was supposed to do, which is to reduce neuroligin 2 expression. So we see that in vivo, 24 hours after injection, neurolide 2 reduces neuroligin 2 expression. And when we look behaviorally at neurolide 2, it reverses the hyperalgesic priming. So if we give it after we've primed animals, in this case, we're priming them with BDNF. If we give it after we've primed the animals, it very strongly reduces the magnitude of the PGE2 response, suggesting that neuroligin 2 is functionally involved in the maintenance of hyperalgesic priming. Not only that, but if we, instead of challenging the animals with PGE2, challenge them with a D1, D5 agonist and disrupt neuroligin 2 function with neurolide 2, that completely reverses the effect that the D1, D5 agonist has. So this suggests that disrupting neuroligin 2 function can reverse this chronic pain state. So I just want to wrap up with some data that doesn't come from our lab at all, that we are very intrigued by, and I think gives us the direction that we're going to go. So in the future, in trying to understand and kind of integrate all the data that I talked about today, So this paper came out in PNAS a couple months ago, and it shows something that is super interesting to us, and that's that neuroligin 2 is expressed in GABAergic neurons and is found in GABAergic synapses, but it's also found in dopaminergic synapses. So, you know, we kind of started this whole story looking at how dopamine may be a novel player in the maintenance of a chronic pain state. We showed that GABA is changing and that this involves neuroligin 2, and that the population of neurons that are changing in the chronic pain state seem to be GABAergic and probably are receiving some dopaminergic input from A11. Well, now we have this evidence. This is from the brain. I think these experiments were all done in hippocampus, but we have some evidence that neuroligin 2 may also regulate dopaminergic synapses. So this is something that we're really interested in now, that maybe this is the kind of integrating factor that causes the circuitry to change to promote chronic pain. And that if we could disrupt this, this would give us a way to alleviate chronic pain without influencing normal acute pain. So we're now, I think, based on all this data, rethinking how the descending modulatory systems are working in the once pain becomes chronic. We've found, as I already have mentioned several times, this unexpected role for dopaminergic modulation of spinal cord circuitry that appears to be coming from the hypothalamus. We're pretty excited about identifying D5 as a potential new target for chronic pain therapeutics, and we're testing this a lot now. So what Salim Magad is really working on in the lab now is what is the phenotype of D5 receptor knockout mice, and we're very interested in the idea of making specific D5 antagonists. We're also hot on the question of trying to determine exactly which neurons these are that are modulated by dopamine. And again, we think that there are these neurons in the medial part of lamina 3 and lamina 4 in the spinal dorsal horn. They may be GABAergic. They may express neuroligin 2. And this whole idea that neuroligin 2 is regulating dopaminergic synapses on GABAergic neurons could be the thing that could kind of help us to tie everything together. So this is the hypothesis we're now currently pursuing. But kind of taking a step back and looking at it from the really big picture, the thing that we're really most excited about is the idea that the circuitry that is involved in acute pain may actually be quite different from the circuitry that's involved in chronic pain. And that provides this really unique opportunity to potentially disrupt chronic pain with disease-modifying therapeutics while leaving acute pain intact. So again, this work was mostly done by Ji-Young Kim, who was an exceptionally talented graduate student in my lab and is now being carried on by Salim Magad. Gallo Mejia is my lab manager and makes all of this possible. And Jamie Moy is a graduate student who's also been working on the project. Greg Dusor has been my long-term collaborator. We worked together on all of these projects. And the Neurolide 2 was all made by Joseph Wagner, who's a peptide chemist at University of Arizona. And the funding was provided by the National Institute of Health and the Rita Allen Foundation. So that's the presentation. Yep.
Can you, okay. Can you unshare the presentation? Okay, great. Yeah, this is an interesting presentation. The story has evolved over time.
Yeah.
Into a kind of like a neuroligin thing. It's like, do you guys have any idea on what is the internal ligand which does all these things?
No. So, I mean, that's something that we'd really like to know. We think that it's possible that the hypothalamus is becoming engaged as pain transitions to a chronic state, and that dopamine is promoting all of this plasticity. But we don't really have a lot of evidence for that. So another thing that we're trying to do now is to use optogenetics to drive the activity of these A11 neurons to see if we can induce a chronic pain state simply by turning these neurons on. So if that were the case, I think that would give us a really good window into how dopamine is involved in changing the circuitry in the spinal cord.
Okay.
And of course, we D5 knockout mice to try to understand what the receptor is, right? So,
yeah.
But in terms of looking at whether neuroligin 2 is being changed by dopamine, we have the neuroligin 2 expression changes are mediated by dopamine. We haven't looked at that yet.
And like Parkinson's, like you have this dopamine issue.
Yeah.
So like, is there any connection? Like, can you extrapolate these results into Parkinson's patients? Do they feel any pain less, more, or like how does it work there?
Yeah, so there actually is a fair amount of evidence that Parkinson's patients develop pain problems.
Mm-hmm.
They have a loss of their substantia nigra dopaminergic neurons.
Yep.
I'm not aware if there are losses of hypothalamic dopaminergic neurons. It's an interesting question. Actually, I've never looked into the literature to see if that happens or not. One thing that I think is kind of emerging in the pain field is that there is plasticity in the dopaminergic system that appears to happen throughout the brain. So, you know, I talked today about the A11 nucleus and projections down into the spinal cord, but there's a lot of work that's been done by Frank Parecha's group at University of Arizona, Daniel Karian at Northwestern University, Irene Tracy at Oxford, showing in animal models and in humans that there are changes in the dopaminergic reward pathway when pain becomes chronic, and that these VTA accumbal projections may be involved in the promotion of especially chronic neuropathic pain. So an idea that we're kind of trying to push is that there's maybe this global dysfunction of dopaminergic signaling in the brain that is involved in pain chronification. And you know, we're excited about being able to manipulate part of it with D5 receptor antagonism. So,
but you don't see any Parkinson's-like symptoms in the pain?
No, we certainly don't. And the lesions that we do, we're very careful to also make sure that we don't have any problems like that, because motor deficits would obviously totally throw off all of our experimental results.
Okay. Yeah. Thank you so much, Dr. Price. It's really nice